What I'd like now to do is to give you an example of a very large-scale genetic epidemiological study in which I'm involved. It's called the Collaborative Study on the Genetics of Alcoholism. It has 6 collection sites that span from Southern California and San Diego across America all the way up to New England, in which the University of Connecticut is the northernmost collection site. This study began in 1989 with really the intent of trying to identify genes that were responsible for the development of alcoholism, as we called it in 1989, alcohol dependence. Currently, the study has been funded by the NIAAA since its inception, and we have received funds from NIDA, the National Institute on Drug Abuse, to help supplement some of the work that we're doing. As you can see from the map, the collection sites do go across from San Diego to St. Louis, Iowa City, Indianapolis, Brooklyn, and then up to Farmington. We do have data analytics sites in Piscataway, New Jersey. We have collaborators from the NIAAA in Washington, D.C., and a very large data analytic group, genetic data analytic group in San Antonio. The COGA study was really designed, as I said, to identify vulnerability genes with a focus and with the knowledge of it, that this, in fact, is a complex disorder. We did take great care in obtaining DNA, but also in developing cell lines that are stored at Rutgers University in New Jersey, so that we, in fact, can go back to these cell lines and get additional DNA as necessary. We also took great care to try and assess environmental variability, both in terms of the home of rearing, the parental home, as well as in the individual's current house, if they are married or living with a partner. It was innovative in that we, in fact, did look at endophenotypes, which are biological-based endophenotypes, mostly related to electrophysiology variables, such as the electroencephalogram or the event-related potentials. We also look at compound phenotypes, whether the individual has alcohol and other substance abuse disorder, alcohol, other substance abuse, and a psychiatric condition such as depression, etc. Because it's very clear that given the heterogeneity of alcohol dependence, that there is not a single phenotype or a single diagnostic category that's going to yield, if you would, good, solid genetic information because of the complexity. As I said, we really do take great care to phenotype the individuals. And with this, we use a standardized diagnostic interview, which we call the COGA. It is aptly named in that it is very time-consuming. It takes probably 60 to 80 minutes to complete with an individual, but it covers all of the Axis 1 disorders in the DSM. We started with 3. It's now compatible with DSM-5. We look for endophenotypes using electrophysiology. This is a good example in that top panel, number 1, of what our families look like. We really do try and get large pedigrees, multi-generational. And if you look at this particular pedigree, you can see that the proband, the very top square there, is darkened. That would be the mother, and in the offspring, 3 of the 5 offspring are affected, and in the one affected individual there also has 2 grandchildren now, and they're affected. This is just a cartoon, but it does give you a good indication of what COGA does, or what any other good genetic epidemiology study would do that is focused on large pedigrees. We then also, our first pass analysis is a linkage analysis to identify regions in the chromosome that show some signal in relation to the phenotype of interest, i.e. alcohol dependence. And once we have those signals, or we've identified regions, we then use association types of analyses, including GWAS, which I mentioned previously, to try and identify specific genes relevant to alcohol dependence. Here in the sample description, you can see the types of families that we have, as well as their distribution by age. We have stage 1 families in which there are only one person in that entire pedigree that's affected. We have stage 3 and 4 families, 2, 3, and 4 families in which multiple individuals are affected, usually 3 or more within the pedigree. We have also established a comparison group of individuals and families that we've recruited from the community using dental clinics, sports teams, drivers and voter registration to solicit families who might be interested in participating. At this point, we have over 17,000 individuals who are involved in the COGA study. And of those 17,000, almost 10,000 are adults, and the remaining are children or adolescents that we're following forward every 2 years to track their development from early adolescence, usually around age 12, through emerging adulthood, through age 30. The gender distribution is very nice. It's almost 50-50 in terms of male-female. And the ethnic racial composition of the sample is relatively close to that of the United States, with Northern Europeans representing about 3/4 of the sample, African Americans about 17% of the sample, Hispanic individuals representing about 6% of the sample. We did not do well in terms of being able to gather information on either Native American or Asian groups, and so consequently what we have done is develop companion studies with, for example, the Japanese in Tokyo, Japan, who provide us information on, at least on Japanese individuals, although they are not totally representative of all Asian populations. Similarly, we have conducted a companion study in Anchorage, Alaska, with Native American groups that provide companion information on those, even though they're not, neither of these two samples are included in the overall COGA study. As I indicated to you, we probably have identified 30 to 35 different genes associated with alcohol dependence. And I think one of the things that's very important to recognize is that the genes we have identified have appeared in some ways in pathways that you would expect. That is neurotransmitter systems such as the GABA system, the muscarinic system, in the dopamine system, etc. And as you can see from this first slide, in fact, many of the genes that we have found are, while related to alcohol consumption or alcohol dependence, are also related to a variety of other traits. And those traits could be related, such as other drug dependencies, behavioral disorder, or potentially to CNS functioning, such that we might see in terms of EEG or event-related oscillations, or even in terms of IQ. It's also important to note in here, and I think one of the things that's quite interesting, is that in fact taste sensitivity also does affect or has an effect on the drinking history of individuals, as well as the potential for developing alcohol dependence. And here we see there are two taste receptor genes, R38 and R16, that in fact are associated with the development of the disorder. as you would guess, individuals who are very sensitive to bitter taste, in fact, do not like to drink beer or many other alcoholic beverages for that matter, and such that if you're homozygous, that is, you have both alleles of the TAS2 R38 gene, the likelihood of developing alcohol dependence is quite low. Take a look now also at the middle and the bottom part of the table here, where genes related to the nicotinic receptors, i.e. smoking behavior, or opioid, such that or may affect opiate dependence, are also related to alcohol dependence. And again, with many individuals with comorbidities, in fact, it's not a surprise that smoking and or opiate use may in fact be related to the course of the illness in individuals with comorbidity. Similarly, as you might expect, the genes related to the metabolism of alcohol dependence also in fact are related to the development of this disorder. ADH, or alcohol dehydrogenase, particularly ADH4, is quite important. As individuals who have both alleles of this, homozygote, in fact do drink a lot, much more than individuals who are heterozygote in that regard. So that it's quite important that how much you drink will, of course, affect the course of the disorder and probably the severity of the disorder. I think that we should keep in mind that ethnicity, in fact, can influence the rate of alcohol dependence, and it's related to biological differences. Alcohol metabolizing genes, and there are two sets. One is the ADH, the alcohol dehydrogenase gene, and the aldehyde dehydrogenase gene do affect how, not only how quickly people metabolize ethanol, but also the effect of the ethanol in terms of the CNS. One of the things we know is that individuals of Asian descent, particularly Japanese and Koreans, who have the ALDH22 gene, have extreme difficulty in being able to metabolize acetaldehyde. It's the first metabolite of ethanol, and this is also very toxic, and individuals with the ADH22 gene in fact end up with the flushing response, where you will see the face turn red, sweating, sometimes nausea, and often individuals will become quite sleepy as well with this particular gene. ADH4, as I mentioned before, is related to how much people can drink and how quickly without feeling an effect. And then we also talked about taste sensitivity as a particular protective factor for individuals who are sensitive to bitter taste.